before you treat with the compound, you ask, where is polymerase? And you find that there's only one gene there, but the polymerase is anchored at the start site. There are a couple others, these are already on, and a compound does not change them that much. And it's only one of them where there's a change in serine 2 levels. This is the phosphorylation that tells you you're going to transcribe through. And so basically what is happening, even though the compound is bound to 250 different places at different levels, it is only acting at that one gene because there's a receptive polymerase waiting for that signal. And as it interacts with our target that we have brought in, BRD4, it can now transcribe through, make that RNA. And so what we've done is by using the same principles that biology uses, fuzzy logic, if you will, we have 250 to 1,000 places where this molecule goes to different degrees. But if you superimpose on that the requirement that what it brings to that site will only work on a polymerase that is sitting, waiting for that signal, right? So you layer another level of specificity on there. And what you get is a single gene specific regulator of a disease gene. And what happens in patients typically, scoliosis is the first signal that something has gone wrong. But typically people die of heart problems. You can sort of see 29 patients, majority of them have cardiomyopathies. They die of ataxias later in life. So I just had a parent in my office on Friday who told me, if you can fit my daughter's heart for another 10 years, I'll worry about the brain later, right? So can we get to the heart? So what the student in my lab did is basically, this is what happens to the heart. So the ventricular space basically gets smaller and smaller as the muscle is trying to pump more. It's building up the muscle while it's squeezing the ventricles, and very little blood goes out. And eventually, this is why the patient really dies. And so these are the guys who did the work. Aska did the work where she took induced pluripotent cells. These are cells that have been taken from the patient, brought back to a stem cell-like state. This is something we can do very easily in Wisconsin because this is where some of the technology was developed. Yamanaka won the Nobel Prize for it. But for humans, this was done in Wisconsin, and we basically have access to those colleagues. And we know the signals to convert those cells into cardiomyocytes. I told you this is one of the things that we do. So she made these cardiomyocytes and then showed for certain that these are cardiomyocytes by doing PCR to make sure that you have extreme sensitivity. In fact, the cells, when they convert to cardiomyocytes, turn on the markers for cardiomyocytes, do not turn on the markers for stem cells. And the cells that we started out with, pluripotent cells, have those markers on and not the cardiomyocytes. So this all looks good. But the cool thing here is that we treat these tissue with our molecule, and frataxin is on. And this is a tissue that's been very hard to turn this gene on using other methods that are out there. The next thing, of course, is the brain. You're losing the cerebellar region, this region that is going to give you your ability to walk, talk, and do all motor functions that you typically do. We've taken same iPSC cells, made them into neurons that represent cerebellar neurons, sensory neurons, and dorsal root ganglia. In each one of those cases, I wouldn't be telling you if the protein wasn't been made. And here, patients with different levels of repeats. This patient has very few repeats. You see that they're making, in neurons, they're making the frataxin gene to some level already. Our compound in this context is not making any more than what you would see under normal circumstances. So it does not go beyond the level that is already prescribed by the promoter. How much ever polymerase you load, that is how much is going to get to the end. You're not going to load more. So there's no danger of making too much. And this was done with Novartis, by the way. And then the other thing that we did, I was describing some of this work earlier, a year ago, to Brahmachari, who sits in IGIB. And I said, I have a colleague here who actually does ataxia screens. He's a doctor at AIMS. Patients are referred to him. He then does PCR for 20 different things and says, you have this kind of disease, this kind of ataxia. So every Friday, he collects 4 or 5 patients who have Friedreich's ataxia. And over 3 weekends, he tested 12 patients with our molecule, basically take half of it to figure out how many repeats they have. And the other half, he treated with our compound. And in each one of those cases, with the exception of this one, which was already making more frataxin than it should have been based on the number of repeats this person has. And so what we think we've done in a rational way, looking at the gene that we're going to target, bringing the machinery that it needs, is fix this aspect of it. What has been done in the field is this. People have tried to use histone deacetylase inhibitors broadly. That does the function to a low level, but it also has huge toxicity issues. People have tried to fix the downstream consequences. Can you use antioxidants, chelators to keep the free-floating iron, because now frataxin does not allow you to make clusters? And all of these things have not quite worked, and what we've done is we've sort of intervened at this level, targeting genomes with molecules that can target a particular sequence, and that's one path on one disease. Now, since we can make a molecule which has a DNA binding domain that you can design for whatever sequence you like, here are 40 other diseases that occur because of these repeats. We have compounds that hit every one of those repeats. And now the question is, what are you going to put on the other end to make them specific for that one disease? And we do believe that there is such a thing as precision-tailored therapeutics. I think that world is here with us today. With that, I should thank these guys who've done all the work, guys who've done all the funding of the work. But I can't resist telling you the second half of the story, how far away I am from my time. I've passed my time 5 minutes ago. Thank you. So I was hoping he would say that, so I can tell you the story properly. This was at the event that was held in Wisconsin in 2009. And it's really nice to be here. I'd like to thank the organizers. Rajendra here, Tom, of course, and Tilak Saab for repeating this. Here's Umesh talking to Dieter. Here's Gobind talking to scholars. Here are some of the scholars of the program. So these are not people who were trained by Gobind, but are Eklavya project like myself that have read Gobind's papers inspired by him. And we hope someday will become like Gobind. This is Nomura, who just iconic, and Nirenberg meeting with Gobind after many, many years. They shared the Nobel Prize for decoding the gene. This is the fellow I spent my time with who taught me to think of molecules as dumbbells. I came from a chemistry background where we looked at every atom to looking at spheres as domains. But in any case, this was the event that we held in 2009. Tom was there. And if you look at all these events, the Memorial Symposium here, the Memorial Symposium at ISC in 2011, you will find that Tom is always there, but yet he is extremely modest. He never points out that he actually is always there with Gobind. So in 2007, I went to meet Gobind, and Tom introduced me to Gobind. I had met him before, but he didn't remember me. And we talked about starting a Khorana program to honor him at Wisconsin, because most of the work that he had done at Wisconsin had really, has been the foundation for modern biotechnology, chemical biology, synthetic biology, etc. All the things that have matured into these new jazz words or buzzwords, he was already doing in the 60s, right? So he set the stage for how you think about these problems and how you bring chemical tools and genomic tools and synthetic tools to understand biology. And so he won the Nobel Prize for this in '68, and next year will be the 50th anniversary. It's also the 10th anniversary of the Khorana program. The person who took this photograph of us is my very first undergraduate, Jay Bradner, who now runs Novartis and has developed these JQ1 molecules, a whole platform, and someday he will win the Nobel Prize, I bet. But what I'd like to tell you a little bit about, if you're not already familiar with it, that this program basically has 3 components to it. It is to provide students experience in a research lab that might transform them in a different cultural setting, to see what the horizons are and whether they can fly in that environment, what they can walk away with. There's rural development. This was brought in by other colleagues, including Gobind, and then, of course, to make it sustainable academia partnerships. The government of India joined in 2009, and if Vijay Raghavan had been here, I'd tell him how much money he gave us. But in addition to that, we had money from the University of Wisconsin to start with, and the labs that have taken on these students. This is what the students experience. They come over the summer for 10 to 12 weeks. Indian students come to labs in the US, leading labs in the US, including many of the people sitting here. And US students come back here and they go to ISC, they go to TIFR, they go to NCBS. But basically they also change their perspective on how the world works and how science is done in different environments, and the fact that creativity, resourcefulness, inventiveness exists in both environments, and how it enables different solution of different problems. Students really benefit from this immensely. But that was originally funded by the DBT, and it was limited to biology, chemistry students. So then DST stepped in and made essentially a BOSE program in 2013. So 5 years after the Khorana program, BOSE program came online. And that does the same sort of thing, but for students who are not biological. All science, math, engineering students are in that, economics as well. This is the whole cohort of students, about 100 of them, at the consulate for the orientation. And once it grew to this size, we had to start a non-profit to run it, because now what we were doing is we were engaging a lot of universities. So students choose the place where they want to go and have that summer internship. We make sure that we find the right mentors. If they have troubles with their mentors, we step in, and it becomes very difficult if it isn't within the 5, 6, 10 universities that you're familiar with. Once it goes to this scale, there isn't any major university that we haven't placed student in, and that they haven't requested students back, including Chicago. So we've done this for students. It is going really well. The nonprofit now has been able to expand the rural development component. We've worked with Vishal Bector at PAU here. We've worked with Aysel Mohali very closely. And now we're doing industrial partners like Mahindra and Mahindra to try and see how we can enable rural development at the village level. But the students, the original batch of students, now Devesh is here somewhere. Devesh, are you here? There's Devesh. He was in the very first batch, 2008. He's finished his PhD, won the best PhD thesis award. He was at Roorkee every single year. He stood first. We are still writing a paper. He's thinking of starting an institute in Jaipur as just someone who's just finished a PhD. But his cohort and cohorts after them have graduated and now finished a postdoc, and they started this organization to try and see if they could enable people who are in the US, you know, to come back to India and establish their careers here, or collaborate in a way that is meaningful, or develop other models where you could actually leverage the strengths of both countries and others. And Arun is here. He showed up. I just saw him. There's Arun. He's just come back as a Ramalinga Fellow. He was there till when? Till last year? Earlier this. So he and others were involved in building this initiative for postdoctoral fellows and senior students. And this was the first time we did it at University of Chicago and the consulate, where this team here from various universities actually came together, held this event, brought 150 people, and I think 20 or 30 speakers from India and from industry, Petroda and others. Vijay was there, etc. So this is now a recurring phenomena. We've done this again in 2017, we'll do it again in 2018. And so let me just tell you a little bit about anniversaries. So that's the report card. We've had the 50th anniversary of the Khurana Nobel Prize, well-deserved Nobel Prize, prize that lifted the nation at a time when we didn't have this. So the impact was much greater than the award for discoveries and inventions that Gobind brought to science. We have the 10th anniversary of the program that we started in his honor to make future Khuranas, right? We have the 5th anniversary of the Bose Scholars Program for people who are like Khurana scholars but not biologically inclined. We have the 3rd anniversary of SciROI type programs, science and research opportunities in India. And what is needed is this. This man So what I'd like to propose is today we launch the project for Uttam Rajbhandari Scholar Fellows to bring people from Nepal and the reason I'm doing it here today is because I've asked him to do this for the last 5 years and he said no. I'm trying to bring peer pressure to bear on him. So you will agree to this, Tom? Here's the team that will help you do it. You don't have to do anything, just say yes. I'll collect money from the first row. Yes?
I'll always think about what you say.
I heard yes. Thank you very much.
That was a splendid talk, absolutely splendid talk. Thank you, Asim. Yeah, so unfortunately we do not have time because there is a poster session going on right now. And so we'll go to the poster session and there's tea. And then we will meet at 12 o'clock here instead of 11, whatever it is, 11:35. So we'll start the next session at 12 o'clock. And Matthew Sachs will chair that session. Okay, thank you.
And